Okay, so today I'm going to be telling you guys a little bit about the coronavirus testing, or as you may know, COVID-19. This testing is fairly new. Before we do the testing, we do need a collected sample from the patient. I do believe they are going up the nose or in the back of the throat. Once we get that sample collected, we will test the sample using real-time PCR. Once the sample is collected, it's treated with several chemical solutions that remove substances such as proteins and fats and extracts only the RNA present in the sample. This extracted RNA is a mix of a person's own genetic material and, if present, the coronavirus. The RNA is reverse transcribed to DNA using a specific enzyme. We then add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. These fragments attach themselves to target sections of the viral DNA if the virus is present in the sample. Some of the added genetic fragments are for building DNA strands during amplification, while the others are the building the DNA and adding marker labels to the strands, which are then used to detect the virus. The mixture is then placed in an real-time PCR machine. The machine cycles through temperatures that heat and cool the mixture to trigger specific chemical reactions that create new identical copies of the target sections of viral DNA. The cycle repeats over and over to continue copying the target sections of viral DNA. Each cycle doubles the previous amount. 2 copies become 4, 4 copies become 8, and so on. A standard real-time PCR setup usually goes through 35 cycles, which means that by the end of the process, around 35 billion new copies are formed of the sections of the viral DNA and are created from each strand of the virus present in the sample. As new copies of the viral DNA sections are built, the marker labels attached to the DNA strands and then release a fluorescent dye, which is measured by the machine's computer and presented in real time on the screen. The computer tracks the amount of fluorescence in the sample after each cycle. When the amount goes over a certain level of fluorescence, this confirms that the virus is present. We also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection. The fewer cycles, the more severe the viral infection is. And I do believe we are most of the time getting these results back within the next day or the next night, depending on how many patient samples we have. But that is just a little bit about what's kind of going on in the lab. Thank you.